Thank you very much. And that's like really kind words. Now I feel a bit of pressure, but I will do my best. But so that as you have heard, I am a physicist, and I don't know the distribution of people in the audience, but if I say something that really doesn't make sense, then like be prepared to ask questions, and I'll be very happy to answer whatever I can. But so that I'm a physicist by training, but I have been working on this, the phage bacteria world, and it's such a fascinating small beings that can do so many things. And every time I learn something about the new finding about the phage and bacteria biology, I'm like so excited. But so the one thing that I'm very impressed by the phages is just this powerful way of them reproducing, so that just infecting one bacterium is enough to create sometimes like hundreds of phages, and that is like very unique thing for the bacteria and the phage. Say that this is kind of, to me, a bit like predator and prey. But if you think about the rabbit and fox, then fox need to eat many rabbits before they reproduce. But for phage, it just need to eat one cell, and then they can create hundreds of them. So that's really powerful predator. But that's also this very big amplification means that even if we deal with like huge number of bacterial cells, just starting one infection is just enough to eventually give a very big impact to kill everyone. So that like this single event becomes the very crucial to describe the what's going on in this population of bacteria and phage. And also that also means that when and where bacteria actually meet the phages matters a lot for how the dynamics goes. So within this talk, I want to specifically think about this where and also when the single event happens in this phage bacteria context. So that I want to talk, consider different effect of the spatial structure can change the bacteria and phage, the interaction, and also how the stochasticity of the event, like when the single phage just by chance hit the one bacterium, could change the outcome of what we see in a big scale. So in the first story, I want to talk about what happens if bacteria form aggregates. And then in the second story, I want to talk about what happens if bacteria can swim. But so let me motivate my first half of the story from the very sort of the classic way for the modeling people to model bacteria and phage population dynamics. So then normally we like to think about the well-mixed system as a simplest model system, things like chemostat. And then if you do the experiment where you put bacteria and then the virulent phage, and then maybe over time, of course, the bacteria get eaten and then population goes down. But then also the phage also goes down and then bacteria recover. And then after some time, they can reach some kind of steady state where you have some amount of bacteria left and then a lot of phage are in the system. And this kind of the system can be understood very well by using the differential equation that think about the bacteria growing exponentially, but then there is a carrying capacity, and then when the bacteria and the phage meet, then they get infected, and then that will result in the burst of the phage, and then the phage also have some dilution rate if it's a chemostat. And then if you try to solve this for the final steady state where the population doesn't change over time, and then you can find a solution. And this final state it's such that the phage start to eat bacteria, and then bacteria population goes so low that even if, say, one burst give you the 100 of phages, the 99 of them will be diluted out before they find the next bacteria. And then basically you can balance. And that's sort of the understanding of this final state. And that is good that the model can also explain these kind of the final state. However, this kind of the simple equation that have several problems because of course it's very, very simple equation. One of the big problem of this kind of equation is that if I start from the starting point which is not in this balanced state, then it will have some transient oscillation of the bacteria and phage population, but this oscillation is normally really high in amplitude. And that's sort of so high that it doesn't really match with what you see in the experiments. And of course there are many many things that we did wrong when building this model, and many of those contribute to this sort of artificial transient oscillation. In the chemostat system, maybe one of the things that is wrong could be that we are treating the phage trait and bacteria trait as a constant. So the old bacteria, for example, have exactly same the absorption rate to the phage. That's probably not true because some bacteria will express the receptor more than other bacteria. And if we sort of put that kind of distribution into consideration, probably there will be less synchronization of the population, and then maybe we will have less the oscillation. Another factor that could even happen in the chemostat could be that the bacteria may form a little bit of the structure along the wall of the chemostat, and then this equation which assumes we can describe everything based on the concentration may become a bit wrong because there can be some difference depending on where in the chemostat you're thinking about this population dynamics. But now that actually I want to leave from this chemostat and then try to think about the bigger scale where we still often use this kind of the concentration-based population dynamics as a way to describe the system. And that can be, for example, in the ocean or some other liquid system. But then in those those aquarium system, these days people are talking about actually the bacteria can still form some kind of structure, so and especially they could form some micro aggregates. And here for example, there are several sort of suggestion of how the aggregation formation can occur. Maybe if bacteria grow, but they stick to each other, then maybe just the growth becomes the formation of the colony. Also, they could be attached to to each other because there is some material that they can stick to. But there are different ways to think about the bacteria actually are not always planktonic, but they could form small aggregates. So we started to think that how does this kind of aggregates can affect to this population dynamics. And first we made a very simple consideration. So let's think about the chance of a phage finding a bacteria. And of course, the phage cannot really swim. So this is done based on the so-called diffusion-based search. Basically, the phage is diffusing randomly, and if it hits the bacteria at the right bacterial surface, then roughly speaking, you can get absorbed. This kind of the analysis has been done a long time ago, and then what people know is that the rate of finding a target, meaning in this case the bacteria, is proportional to the diffusion constant, but also proportional to the radius of the target. So roughly speaking, if I have the bacteria of the radius A, then the absorption rate is proportional to this A and then diffusion constant, and then there is some theoretical constant in front if it's a sphere. And then if you have say B bacteria, say B is a number, and then in the system, and if it's well mixed, then rate of finding one of them would be this eta times B, because there are B of them. And then I want to compare this with a situation where these B bacteria is actually not swimming freely, but they form an aggregate. So that if you let them make a aggregate, then so let me imagine that they make a very dense aggregate. So that I put them into a ball. Then this entire ball has a radius of R, and that ball should contain all the B bacteria. So the volume of this ball should be proportional to the number of bacteria I have. So roughly speaking, what I expect is that the radius of this ball should be proportional to B to the 1/3, because volume is R to the cube. So that means that the chance for the phage to find this ball goes the original this eta per bacterium times B to the 1/3. So what this means is that if the bacteria make a ball, then finding the first cell for phage becomes harder. It gets slower when the bacteria makes an aggregate. But of course, once the phage find this ball, then burst over there, then they are now in the feast, right? They are all the other bacterium next to it, so that it's very easy to find the next bacteria. So that's kind of the trade-off between the making the aggregate and then the harder to find, but once you are found, then you are all found. So we started to think about how this would play out in the population dynamics, and we decided to make a simplest possible model I can think of. So and this was done by our PhD student Anjali, and the model is like this. So that we just assume that single bacterium, when they grow, which they grow exponentially, they form a colony. And then I consider a system where there is a bit of the influx of this new bacterium, and then they are coming in, and then they will grow and then make the spherical colony over time, and then so that they grow over time. But in the system, there are also phages. And then these phage are floating in the system, and then they will find this colony at the rate proportional to this colony size, which is the number of the bacterium in this colony to the 1/3, which grows exponentially. And then once this colony is found, then of course in reality there will be it will burst, and then there's next phage, the infection, and then it will burst, and then there will be those sort of the recurrent infection to kill all the cells in the colony. But for now, for simplicity, I say that I just sort of say that once you are found, then everyone is eventually infected and then give birth of many phages. And then the number of phages that comes out is proportional to the number of the bacterium in this colony. And then I just try to simulate what happens when you have this kind of the very simple model. And then let me compare the outcome first in terms of the total population, when we consider aggregates and when we don't consider aggregates. When we don't consider aggregates, but then we have this the flux of the bacterium coming into the system, but then they float, they grow, but then they become single cells. Then we get this very sustained oscillation. But when we make the aggregates, then what we see is that total population number stabilizes to the constant value when we consider large enough volume. We can show what happens in such a situation. If I can click stuff, can I click stuff? Yes. So what we are trying to show is that all these dots are expressing the size of the colony in the system, and then it's a little bit sort of the misleading in the sense that these colonies are not moving in the movie. It should be actually in the 3D, and then they are moving around so that they are actually well mixed system. But that will make the movie too confusing to see. So that here, it's just to show that there are different sized colony in the system, and then they are popping at the different timing in a very stochastic manner. So then what you are seeing on the top is the number of the phages, and then also the number of the colonies in the system. So then what you see is that when the big colony pops, then there will be the sort of burst of the phage, so the phage number goes up, and then that will then attack the other, then the chance of the other colonies get attacked by phage, of course, goes up. And then there is this local burst boom of the phages, and then that will sort of remove this synchronized population oscillation, because this finding the colony is very stochastic events. And therefore, when you average over the population in the big volume, you get this the constant total population. Another thing that we observe in this situation is that when we try to plot the aggregate size distribution, then we see that there is kind of parallel of the distribution for the small aggregates, and then there is a cutoff in the large aggregates. And this is because of course the bigger the aggregate is, the higher the chance to be found, because absorption rate is proportional to the bacterium, the colony size, to the 1/3. So there is a punishment to become too big, and that's sort of the make this sort of the colony size limited by certain size. And then how this distribution look like will be dependent on the influx rate of the new bacterium, which is parameterized as alpha in this system. And then so that if you don't have, if you have the very high rate of the new bacterium coming in, then there will be the many colonies, but that's also the increased amount of phage. And then that's prevent the colony to become very, very big. And then when you have a rare events of the new bacterium comes in, then you have sort of a bit less the bacterium to support the phage. So the phage number goes down a little bit, and then that allows the colony to grow a bit bigger. So therefore there is difference in this, where is the cutoff. But also this sort of gives us a little bit of funny relation between the influx of bacteria and the total population. So when we increase the influx of bacteria, then that will of course increase the number of aggregates. But then that will sort of and that will increase phage a little bit. But then the average aggregate size goes down. So therefore, actually the total number of bacteria does not change that much when I change. At least it's a lot. It increases, but a lot slower than linear with the influx of bacteria. And then we can calculate that it will be the influx to the 1/4. That will be the relationship. But so there is kind of the self-organized the structure in the number and the size of the aggregate to sustain relatively stable coexistence of the bacteria and phage population when the influx is varied. So this is the very, very simple model of the effect of the aggregates that could happen if we have aggregates, and what it could do in the population dynamics, and it will change the behavior quite a lot from the situation where all the cells are free. And if you're interested, then there is a bit more result in the paper that is published in the Physical Review Letters. I also want to quickly mention that this situation where we assume all the colonies are eventually eaten by all the phage, that is the varied if the colony size or the aggregate size is relatively small. We also previously analyzed what could happen if the colony becomes very, very big. And then we also simulated this ball of the colony attacked from the outside by the many, many phages. Oops, what happened? Can I start this video? Maybe I cannot start the video. If I, yes. So if the small colony is attacked from the outside, then the colony will be basically eaten up. But if the phage attacks the big colony, but still it's still small enough that the nutrients support the growth inside, then sometimes there can be this funny effect of the surface is dying, but inside is growing, and then the overall the colony might survive the phage attack. So if you're interested in this, what happens in the bigger colony, then there is also this reference over there. But so this was my first story about what happens if the bacteria form aggregates. And then now I want to quickly talk about the different story where what if the bacteria swim. And this work was motivated by the previous beautiful experimental and theoretical work by the people in the University of Illinois, and also the Kim Sneppen in our institute was one of the collaborators in this work. So what people did for this experiment is that you mix bacteria and phage in a droplet, and then you inoculate it in the middle of the swimming agar plate. And then, of course, if it were only bacterium, then you know that the bacteria grow and swim out, and then you will see the expansion of the population towards the outside. But then the question is what happens if you mix the phage to it. And also one interesting information to have is that phage diffusion constant is 0.04 millimeters squared per hour, while bacteria's chemotaxis speed is 0.36 centimeter per hour for this specific system. So basically bacteria swim a lot faster than phage diffusion. So if I look at only this, then it sounds like probably then the bacteria can just run away from the phage. And then this is one of the video from that work, and then you see the population of the bacteria as a white cloud. But then inside you do see the zone that the bacteria killed, which appears transparent. But then you do see kind of the phage is doing pretty well, at least stochastically, to follow some part of the bacterium that is swimming out. And this killing pattern, especially how far they reach, is actually sort of the too far to be explained by just considering this phage diffusion coefficient. If phage can only diffuse, then the killing zone should be just in the middle. So this paper actually did the beautiful experiment of the changing initial phage inoculation and also the changing the absorption rate by changing the calcium concentration. And then they saw this all different patterns. And then they also proposed a mathematical model based on the partial differential equation to explain the overall pattern. What they do see is that when you don't have that much phages, then the bacteria do survive. While if you have a lot of phage, then the bacteria got killed, and then the one you see in the middle is actually resistant mutant. But then another thing that they concluded is that to understand this phenomenon, they have to assume that the bacteria that are infected but not yet lysed are still swimming. So basically bacteria need to hitchhike with the swimming, phage need to hitchhike with the swimming bacteria so that they can actually catch, like almost catching up with this the wave of the chemotaxing bacteria. So that is very interesting sort of the conclusion to think about these phages are actually carried by the swimming bacteria. But then when we saw this modeling, it was mainly focused on the overall this patterns of the where is the front of the killing. But some of the experiment had this very, very stochastic the pattern where the killing is not uniform, but more like there is the line of killing stretching out. And that also looked very sort of the random than the periodic oscillation or anything. So that we started to think that what is this cause of this very stochastic pattern? And this stochastic pattern is seen when especially in between where the host is surviving a lot, and then there will be the flip to the virus is killing a lot if you change the parameters, and especially in between in the diagonal of these kind of the diagram, they saw the stochastic pattern. So to understand to think about some kind of mathematical model that could take into account the stochasticity of this system, our PhD student Laura decided to model this system not based on the concentration, but based on the discrete population. So I don't give the very thorough introduction of the model, but in short, it's a kind of lattice model. And then in each lattice site, we have an integer number of the bacteria and phage. And then each of them will move to the next lattice site by the stochastic the rate equations. And then also the infection and the burst happens at the stochastic rate. So basically, roughly speaking, it's the same model as their original partial differential equation, but now that we are treating it as a discrete entity and not the concentration. If we simulate this stochastic model at some specific parameter, then this is what we see in terms of the total bacteria. And here we are showing the total amount of bacteria in the system, infected or uninfected, and then the scaling the color based on the maximum population so that you can see. But so you can see that there is a growing swimming front, the spreading, but then the inside there is this front where the bacteria is being killed by phage. But then you also see some sort of the stochastic line of killing coming into this yellow high bacteria region. And this pattern is actually driven by the occasional swimming of the single infected bacterium going a little bit far away than usual and then bursting over there. So here in the left, we are seeing the same picture as before. In the middle, we are showing the infected bacteria, but the color is in log scale. So that when you see the dark dot, basically it means there is one infected bacteria over there. And then you can see that all this, the sort of the spike of killing zone is led by the single dot of these infected bacteria, which eventually give burst of the phages, and then that will lead the extension of killing zone. And also when we did the parameter sweep, this kind of stochastic pattern happens when the bacteria survival and the virus takeover switches in between. And this kind of the stochastic line of killing is sort of the manifest of the impact of the rare events. And then to test how important it is to have this rare event, the one simulation we did was that we considered the system with the same concentration but different depths. So the meaning that at each location, when depth is thin, then it's the same concentration, but the absolute number of bacteria and phage are less. And then when the depth is deep, then the same concentration, but absolute number of the bacteria and the phage at a given location is more. And then when that depth is shallow, then we have this rare event of the single burst that kills big fraction of the bacteria in the system at the spatial point. So then we get very big contrast of the population of the being killed or not killed. But then the number of the spikes is not many because there's not many rare events. When we have a lot of the bacteria and phage by having the system deeper, then we have more often this rare event. So we have this more of this line of killing coming out, but the impact of each killing becomes less because the one burst kills relatively small fraction of the population. So that as the population goes up, the events averages out, and then pattern becomes smoother and smoother. So that we think that this pattern is purely the stochastic effect because of this demographic noise due to the finite number of the bacteria and phage. So that is the second part of the what swimming bacteria could do as a pattern. And the overall, the main message is that we believe physical structure and stochasticity matters a lot in the phage bacteria system. And I hope that that kind of statement might trigger something new idea in your phage bacteria research. Thank you very much.